Good evening to you all at home. It's my great pleasure to welcome you to the latest of our Bootham Talks, and it's a particular pleasure this evening to welcome our guest speaker, who is a parent at Bootham School and somebody I've known for a number of years now. Simon Smale is joining us this evening. He is currently, he said, getting his information on the screen in front of him, consultant gastroenterologist and clinical director for Manchester University NHS Foundation Trust. Having trained in Newcastle, he worked in London and then had his first consultant appointment in York, where I'm interested to read he won the York Press Healthcare Hero Award for providing excellence in patient care. What a great endorsement that clearly is. His interests, and if you've just eaten your supper, you may not necessarily want to hear all of this. His interests are particularly in functional gastroenterology, IBS, the microbiome, wait for it, fecal transplant, and C. difficile-related diarrhea. So if you've ever wanted to know anything about those things, Simon is your man, and he is speaking to us this evening on the subject of our bugs, how they influence health and disease. It's my great pleasure to welcome to talk to us this evening, Simon Smale. Thank you, Simon.
Thanks very much, Chris. So this is going to be a whistle-stop tour of the microbiome. I'm going to talk about the microbiome in general. I'm going to talk about how we get our microbiome, and then I'm going to talk a bit about the impact that the microbiome has on health and disease. So our bugs, the microbiome, what does it all mean? So it's a collective term, the microbiome, for the bugs that live with us. They include fungi, things like athlete's foot, bacteria, viruses, and protozoa. They live on our skin, and different bits of skin have different bugs living on them. In our noses, our mouths, and in our bowels. But most of the organisms that live with us are in our large bowel, and we have roughly 10 trillion bacteria, protozoa, but basically largely bacteria, living in our large bowels. There are approximately 10 times as many bacterial cells from other organisms within our bodies as there are human cells. And there are roughly, get this, 3.3 million genes within our microbiome compared to the 23,000 genes that we have in our human genome. So there's roughly 100 times more genetic diversity in our microbiome than there is in our human genome. So whereas humans generally share roughly 99.9% of their DNA with other humans, they only share perhaps 10 to 20% of their microbial DNA. So we all have a much more different microbiome than we do a human DNA. And that might explain why some of us, even in identical twins, choose to do different things, have different appearances. And the effects of the microbiome and of these plethora of organisms largely relate to the metabolic products that they produce and the effects that these products have on our human metabolism. So the hope for pharmaceutical industry, at any rate, is that they can identify these products and then sell them to us. I'm not necessarily a fan of that. So the microbiome is huge. So you as biologists will know that things in the animal and plant kingdom are classified thus. So you have a kingdom, animal, plant, and then you go down to phyla, and then genus, and species, and strain. So our kingdom's animal, our phyla is vertebrate. That's a big thing, vertebrates. We're a lot of us. Our genus is mammal, and we are Homo sapiens. So just to give you an idea, there are some animal in their Latin phyla, and then in the English next door to them is what they actually are. So those are the main phyla in the animal kingdom. There are a host of others as well, actually, but we're vertebrates down the bottom there. So to give you an idea of the diversity of the microbiome, these are a rough kind of pie chart of the phyla, big classes of organisms that you might expect to be represented in a typical colon. It'll depend upon what people are eating. It'll depend on what their mums had. We'll come back to that, but it's just to show you that there's a whole ecosystem within our microbiome that we are only just coming to grips with. We're only just coming to grips with it because we're only just getting to the stage where we can understand the DNA, sequence the DNA, and understand the computational biology. To give you an idea, when I qualified only yesterday, it took a year and £2 billion to sequence one human genome. Now you can do it for about £200 overnight. And that's how DNA sequencing has moved on in 25, 30 years. So a bit more about the microbiome and the effects of these different organisms on our general well-being and on the way we function. So we look at bacterial diversity, which is said to be a good thing in the microbiome, at the level of phyla and genus. And viruses, bacteria, and fungi have effects which are species or even strain specific. So although we will look at phyla when we look at the microbiome generally and look at the diversity amongst different phyla, actually the effects are about the strain and actually relate to the product or the metabolic space that that strain occupies. And those products of that bacteria or that organism essentially manifest their effects through their impact on our human cells. So for example, short chain fatty acids, some of which are produced by bacteria in our large bowels, actually have profound impacts potentially on the way our nervous system develops and works. And we'll come back to that. Changes in the proportion of phyla and genus are likely to be markers for the effects on specific strains. So when people say, oh, it's good to have more lactobacilli and bifidobacterium, that's probably true, but those changes probably reflect benefits of particular species within those great big groups. So a parallel might be for humans, if you see what I mean, that we see more bubonic plague when there are more mammals. But mammals are really just a surrogate, a marker for the brown rat in human beings. And the brown rat carries the flea that then bites the human. You could see the flea as the metabolic product of the brown rat, I suppose. So I'm going to talk a bit more now about the development of the microbiome, because actually the microbiome is key to human development. So I'm going to talk a bit about how you acquire your microbiome, perhaps in utero a little bit, although there's a great deal of controversy in the microbiome world about that, in early life and in delivery, a bit about adolescence and what happens then, a bit about the impact of diet and lifestyle. We'll come later to medical intervention and then a bit more on old age. So microbiome and human development, your mum really matters. Much of your microbial diversity comes from your mum, and many of your bugs will have been your mother's bugs. So diversity is thought to be a good thing. If you've got more genes, you've got more metabolic tricks, and therefore you've got more stuff that you can do with the residue that's left when it gets to your colon. And that has a potential to have good effects largely on your health and well-being. So how do you get your microbiome? Well, it starts as you're coming down the vaginal canal, as you're having a normal vaginal delivery. So 99.9% of the bacteria in the vaginal canal, as you're being delivered, are lactobacilli. And as the fetus is coming down the canal, it'll be swallowing, and some of those bugs in the vaginal canal will get swallowed. They'll go through the stomach, into the small bowel, into the large bowel, and they will colonize the large bowel. So that's where the biome begins. And then, having colonized the large bowel, they flourish on lactose. And of course, biologists will know that one of the major sugars in breast milk is lactose. And so they get fed the very food that they desire. And this then, together with the breast milk, creates the perfect environment for things like bifidobacteria, which then metabolize lactose to produce lactic acid. setting up a slightly acid environment. The interesting thing about bifidobacteria is they create lactic acid, and then once the environment becomes too acidic, they don't reproduce anymore. So it's about balancing that ecology, as it were. And then over the first year of life, babies develop a growing diversity of bacteria, depending on a large part on their mother's biome and to a lesser extent on what they're eating and their environment. So by the age of about 3, most of us have got our biomes largely established. So usually microbial diversity within an individual is most variable within the first 3 years of life. It changes an awful lot during that period. Most of it, that change in the first year, in fact. So small children become larger children, and then they become adolescents. And adolescence, the Romans used to say, was any time between the age of about 12 and 25. Sometimes my wife still thinks I'm adolescent, but that's a different story. So as humans mature, the individual biome becomes more fixed, although diversity within an individual sample, an individual person, might continue to increase until you come to the end of your adolescence. Now, quite where that is anybody's argument. You will not be surprised to know that boys and girls are different, and so are their biomes. And the girls won't be surprised to know that females develop greater diversity at an earlier age. Some would say that's why they're cleverer. And presumably that's thought to relate to the influence of neurohormonal mechanisms and things like estrogens and progesterones on the gut microbes themselves. Males often don't develop the same levels of diversity as females until they reach their early 40s, which might explain why we're not as clever, perhaps. So diet. How does diet influence the biome? Well, different diets enable different classes of bacteria to thrive. And this is important because the kind of bugs that you get and the diversity of your bugs is impacted by what you eat. So you get the bugs, a range of bugs from largely your mother and your environment. Interestingly, if you've got a dog or a pet in early childhood, that also increases the diversity of your biome, presumably because you and the pet are sharing things that you then put into your mouth. Whilst there is considerable overlap and a great deal of complexity, 3 enterotypes, in other words, 3 typical biomes, are described. Now, the concept of having a particular type of biome has actually kind of somewhat died a death. So I'm telling you about something that's already gone out of fashion. So 10 years ago, there was this idea that some people grew Prevotella, some people grew predominantly Bacteroides, and some people grew Ruminococcus. And actually, it turns out there's a considerably more overlap in these than was it at first envisaged. And so it's more like colours of the rainbow than discrete groups. And you can shift from one to the other depending upon what you're eating. But in general, people eating a diet rich in carbohydrates will have a lot of Prevotella as a predominant class of bugs. People who are eating a meat-rich diet, lots of protein, lots of amino acids, will have lots of Bacteroides. And people who are eating a mixture of lots of vegetables, and lots of vegetables is critical. So the more vegetables, the better, and the more variability in vegetables, the better. Then you're more likely to have Ruminococcus. And different foodstuffs may encourage the development of very small numbers of different species. And the numbers of the species may not be the important thing. It may just be the fact that the species is there. So a small number of a species that's really important might actually have a big impact. And of course, different things, different foodstuffs have different impacts. And even within a foodstuff, a single foodstuff, different. So, for example, there's an interesting study where they took rosemary at different times of the year and different bugs grow in the context of different bits of the rosemary plant at different times of the year. So who knows how complex the interaction is between your diet and your biome. But eating a varied diet with lots of different vegetables, particularly vegetables are good for diversity, enables your bugs to have effectively a feast of lots and lots of different things. So the predominance of a particular group of bugs, so the three main groups that you see generally, Prevotella, Bacteroides, Ruminococcus, can change with time and depending upon diet and environment. Interestingly, if children are fed on a high fat diet, it reduces their bacterial diversity. And even when they go back to eating a more varied diet, that bacterial diversity remains reduced for at least 6 years. And I say at least 6 years because it hasn't been studied for any longer than that, but it's likely to go on for longer than that, I suspect. So this is why early childhood deprivation and early childhood choices may be so important in subsequent health outcomes later in life. We should pay heed to that. So in terms of lifestyle, sleep, stress, and exercise all impact the biome. Broadly, a broad childhood diet, lots of different vegetables, according to Tim Spector, who's a epidemiologist with a fantastic interest in this at King's College in London, and exercise can have a very positive effect on diversity. And a study by the group in Cork, where approximately a third of the world literature on the biome comes from, showed that in Irish elite athletes had on average twice the number of phyla. So don't forget, that's a big group of organisms in their biomes compared to age-matched controls from the same geography. So somehow it's not clear whether that's chicken or egg. It's not clear whether the biome makes you into the athlete or whether the athletes are living a life of virtue, eating all the right stuff, doing exercise, and therefore have a greater diversity. It's probably a bit of both. So unfortunately, age comes to all of us unless we're unlucky. And as age advances, diversity tends to reduce once you get over about 40. We're not certain whether this is inevitable, and it may relate to reduced exercise because people tend to do less exercise as they get older. And it may relate to reduced dietary diversity. So people tend to be less adventurous in their diets as they grow into their later years. So they'll tend to eat similar kinds of foods. And that may be impacting the biome. We don't really know that yet. It's going to be an interesting computational biology person that evolves, or people that evolve some of the answers for that. It's interesting to note that people who are elderly living in different settings also tend to have varying degrees of diversity. So people living in the community independently have greater diversity than people living in care homes, who are generally more able and have a greater diversity than people living in nursing homes, who again are more able and have greater diversity than those living in hospital. And again, that's not clear whether that's chicken or egg, but it's probably a bit of the two. So what else matters and why does it matter? I'm going to come back to drugs and medical intervention, but I'm now going to move on to talk about the biome in health and disease. And so I'll talk about normal development and how the biome impacts normal development. And then I'll talk a bit about some of the illnesses, but I'll use some examples rather than trying to tell you about every illness under the sun. So I'm going to talk a bit about early life. I'm going to talk a bit about adolescence and some of the diseases people will see in adolescence that some of you guys will be familiar with. I'm going to talk about recent life events, a little bit about the impact of diet and lifestyle, although less so, and one or two diseases that people see in old age. And I'm going to talk about some of the impact of biome on immune function, on neurobiology, which is basically how your nerves work and develop, on behavior and cognition, which is a consequence really of how your nerves work and develop, a bit about appetite and metabolism, and bowel disease, because I'm a gastroenterologist, so I wouldn't be happy without talking about bowels in some way. So I don't know if you guys know about the layout of the central nervous system, but it's not dissimilar from the peripheral nervous system. There are neurons, which are basically the cells that generate electrical activity and send it down the wires, which come out of the neurons and are termed as axons. And those neurons are basically protected, both in terms of insulated and in terms of protected by the central nervous immune system by cells called glial cells. And there are different kinds of glial cells, but the ones I want people to have a little bit of attention to are the microglial cells down here, which look a little bit like, I guess, a bit like crabs or starfish with lots and lots of little branches. And those branches are important because they enable glial cells to communicate not only with each other, but also to allow interneuronal communication, even when the neurons aren't connected by axons. So, interestingly, talking about neurobiology and the impact of the microbiome on the development of the central nervous system. There is good experimental evidence, and it probably happens in humans too, that the offspring of mother rats fed on high-fat diets, so the mothers are fed on high-fat diets, develop problems with glial cell development. So that beautiful stellate, kind of star-shaped, multiple branching microglial cell doesn't develop the branches. It develops lots of little stubby branches, but they never really grow properly. And unfortunately, this impacts the behavior and happiness of rats. And you might say, well, how do you measure the happiness of rats? There are some very elegant experiments about measuring happiness in rats, but essentially, put simply, they give a rat a choice. The rat can either return to a little box and stay in the little box and never go out, or it can go out in the playground and play with all the other rats, or play on the wheel and on the climbing apparatus. And the happy rats are out playing, and the sad rats are in the box. It's slightly simplistic, but there are some good models and parallels with human beings. Interestingly, you can improve the behavior of those rats by feeding them stools, either from other happy rats, and that might sound awful, but rats actually do eat stools from other rats, or happy humans, interestingly. And the same is true of mammals kept in germ-free environments. Their glial cells do not develop properly. Microglial cells particularly don't form those wonderful branch-shaped forms. And associated with this, they develop behaviors of depression, anxiety, and socialization difficulties. And if you then feed these germ-free animals stools from healthy mammals, or happy humans even, the glial cells recover partially. So they develop some of their branching, but they never get back to normal. And the behavior and mental health of those animals improves. And of course, this may have forever implications for humans who are neglected in early life. And one of the things we know about humans that are neglected in early life is one of the things they don't develop is the neuronal systems that support the reward pathways. So whereas when I was young, if I did something good, my parents would say, well done, have a whatever, and I would therefore be happy, and I'd be encouraged to do more stuff that was positive. Kids who are neglected in childhood, particularly in early childhood, as their brains are developing, when in the first few years they're growing massively, they don't develop those pathways, and so they don't get the positive feedback. You can give them positive feedback, but even when they're adults, they don't get the positive warm feelings that other people get from positive feedback, which may explain why even when they're adults, people who've had really poor early lives still don't thrive and respond perhaps how we might expect, although some do, some escape. So I'm going to talk a bit about autoimmunity now, moving on from neurobiology. So as frightening. I know that some people in the audience may have been delivered by cesarean, and you shouldn't worry about this. But delivery by cesarean section, rather than down that vaginal canal where all those lovely lactobacilli are sitting and waiting, and as you come down the vaginal canal, you're sucking up those lactobacilli and they're going to grow in your large bowel. Delivery by cesarean section means that instead of acquiring the lactobacilli over the normal vaginal canal, the fetus gets the surgeon's bugs, so staphylococci and skin flora. And this has future implications for immune regulation. So we know that disrupting mother to newborn transmission of bacteria by C-section delivery may increase the risk of celiac disease, of asthma, and of type 1 diabetes. Now, all of those are autoimmune diseases. And if people want to do some further reading, I have put the references just down here. So it also impacts potentially metabolic disease. So delivery by cesarean section is associated with an increased risk of obesity. And you might say, well, that might just be the fact they've been delivered by cesarean section rather than because they've been impacted by the difference in their microbiome, and that's fair. But it appears to be the bugs. So it's interesting to note that similarly, if antibiotics are given during pregnancy, they also appear to increase the risk of childhood obesity. And one study has demonstrated an 84% increased risk of obesity in children whose mothers are exposed to antibiotics in the 2nd and 3rd trimester. And again, I'll put the references for those publications down at the bottom of the page. I would generally ask, does anybody know what this is? Well, this is a picture of a zombie ant. So zombie ants are ants that have been infected by a particular fungus. And the fungus grows inside the ant and largely destroys it. But one of the things that a zombie ant the fungus does is it makes the zombie ant leave the colony. And it makes the zombie ant leave the colony and climb to a particular height where it makes the zombie ant bite onto the back of a leaf, as this one has done, and basically hold tight where the fungus then kills the ant and dies. And the leaves are all at a particular height, about 12 inches above the ground, which is the perfect height for the fungus to then sprout this rather bulbous looking thing out of its body or out of its head, and for the fungus then to let loose its spores, which float on the air across where all the ants in the colony are going, and then it's able to infect more ants. And so if a fungus, just one fungus, can influence the behavior of an ant, then surely it can influence the behavior of our adolescents. And I'm just going to say a little bit about resilience in adolescents, because I know that anxiety in young people, I think in everybody, pandemic hasn't helped, is a huge issue, but so is resilience. You know, the world is a tough place, and young people are meant to be the ones taking risks. Don't let your parents don't tell your parents I said that, but you are. Young people are the people who are meant to be taking risks, particularly young men. That's what they're designed to do, because in the jungle, we want the young guys to go out exploring, learning new behaviors, learning new tricks, and innovating. And that may explain why PTSD is commoner in young men leading groups, because actually one of the things you need to do if you're a young man leading a group is remember not to take your people in future into a place where you got attacked, which is why you might develop PTSD. So young people are also the innovators. If you look at people winning Nobel Prizes, people almost always do their best work before they're 30. They're adolescents. But the thing is, doing all this is scary, and young people naturally get anxious. Now, there is now a on the market strain of bacteria, a bifidobacterium as it happens, that has been demonstrated to increase resilience, increase vitality, and reduce fatigue in normal individuals, in athletes, and in people with anxiety. And I'd say, should we be giving this to everybody, but particularly our young people who face such challenges? Oh, they have to live with me. My young people have to live with me. So MRI, so there's an imaging technique called functional MRI, which allows you to see which parts of the brain are getting increased blood flow. And we have demonstrated on functional MRI imaging that there are neurological correlates, so changes in the blood flow associated with taking these antibiotics that also correlate with, taking these probiotics that also correlate with increases in resilience in people with anxiety and without. So a bit about metabolic disease in adolescents and in adults. So appetite and metabolism are an interesting thing. So people often look at people who are overweight and they go, it's their fault, they're eating too much. And to an extent, it is about what you eat. It is about caloric intake and what you choose to eat. But there are 95 genes, human genes, that influence your risk of obesity. And they influence two things. They influence your response to food, which is, oh, Simon likes chocolate, he'll have some of that. Simon likes biscuits, he'll have some of that. Simon likes crisps, he'll have some of that. And I like fish and chips, I'll have some of that. Some people are programmed to feel all of those things. I'm one of them. But some people, my wife, for example, aren't. They can see crisps and fish and chips and not think, oh, yeah, fish and chips, I'll have some of those. That's a wiring issue, which is genetic, and it's about the human genome. So obesity isn't necessarily an individual's fault. It is partly environmental, because currently we have so many calories available that we're not fighting for calories, and therefore we can actually pick and choose calories. And the other thing the genes influence is satiety. So actually they make some people get fuller sooner. And if you don't get full, you eat more, and then you put on more weight. But the interesting thing is that the microbiome might influence whether these 95 or so genes are switched on or off. So obesity and obesity-related metabolic disorders are characterized by specific alterations in the composition and function of the human gut microbiome. And there is some interesting thoughts that the manipulation of the microbiome might offer the hope of promoting weight loss and preventing obesity. And there's some interesting stuff going on in America at the moment, where people have had fecal transplant to cure, or to try and cure, other diseases, which I'm going to come to, C. difficile, and they're being sued because they had a fecal transplant from someone with obesity and subsequently became obese, and are suing their physicians because they've received basically stools and a microbiome which switched on the genes that then made them eat more. I think it's still a bit tenuous, but it's an interesting idea. So the microbiome might, through its interaction with human genes and the microbial products that the microbiome produces, may be able to regulate both appetite and satiety. Interesting stuff. Conversely, anorexia nervosa, which is the third most common chronic disease in adolescents, and of all the psychiatric illnesses and mental illnesses, has the highest mortality of all those disorders, is also associated with well-defined changes in the microbiome. So just as obesity may benefit from intervention, so might anorexia. Now, it's interesting to note that similar organisms that appear to be associated with the development of anorexia are also associated with a lower BMI in general, even in healthier adults. Their proportions differ. So this might all be about the interaction between your human genome and its predisposition, your microbiome and which genes it's switching on and off, and your environment, which is all about the images you see and the expectation around you, and then what sports you do and whether you get into ballet and all of those kind of things. So as far as we can tell, as yet, no one organism is causative. And it's more about the metabolic space that those organisms are likely to be occupying and what they're driving in terms of appetite. We don't know the answers yet, but it's interesting. Hopefully we'll get to them. So I'm going to tell an interesting kind of sciencey story about a Chinese herbal remedy now relating to obesity. So for many centuries, Chinese medicine has promoted a particular fungus for weight loss. And some Chinese doctors thought it would be a clever idea to see if they could identify what and why this fungus seemed to make people lose weight. So They discovered that the fungus promotes the growth of a particular bacterial strain, which is then able to translocate across the bowel wall and infiltrate brown fat cells. And within brown fat cells, this bacteria is able to produce metabolic products that uncouple mitochondrial fatty acid oxidation from production of ATP. Now, as you biologists will know, when you do that, effectively you just generate heat, and so effectively you are enabling energy expenditure through heat production without having to actually get off your bums and do any exercise to burn your calories. And so they postulated that you could potentially put the fungus together with the probiotic, the bacteria, and give that to people to promote further weight loss. An interesting idea. But it might explain why the fungus works to promote weight loss. So I'm going to move to old age. I've just got a couple more slides around old age and antibiotics, medical intervention again. And then I will invite some questions. So the gut microbiota is emerging as an important modulator of neurodegenerative diseases. And in one study, enrichment of various genera Lactobacilli, Akkermansia, and Bifidobacteria, and depletion of bacteria belonging to Lachnospiraceae family and Faeculobacteria genus. Basically, those two species are important in generating short-chain fatty acid. And as I mentioned at the start, short-chain fatty acid we know are important in neural development, because we think that may be one of the things that is prevented in the early life if you don't have a biome established, are emerging as consistent Parkinson's disease microbiome alterations. And those short-chain fatty acids, as I mentioned, are important in reducing inflammation and have a critical role to play in the development and maintenance of the central nervous system. And it's thought that dysbiosis, which basically just means a mess up of your microbiome, might result in a pro-inflammatory state, which could be linked to the recurrent GI symptoms, also affecting Parkinson's patients, as well as their brain disease. Antibiotics in the gut. I'm just going to say a little bit about Clostridium difficile related diarrhea, and then I will conclude. So C. difficile is an organism that about 1 to 5% of us, perhaps 20% of healthcare workers, carries without any ailment. It can produce a nasty toxin that causes diarrhea and carries a risk of mortality, if you get that overgrowth of C. difficile, of somewhere between 6 and 40%, depending on which population you're looking at. C. difficile related diarrhea not exclusively, but generally occurs in elderly patients exposed to antibiotics. Now, when you have antibiotics, that typically causes a dramatic reduction in bacterial diversity. So if most of us have 1,000 species, roughly 750 to 1,500, it will roughly halve the number of species in our guts. However, when you stop the antibiotics, most of those species will return within a month. But every time you have a course of antibiotics, you'll lose somewhere between 5 to 10 species forever. Think about that. What does that mean? So C. difficile related diarrhea only arises when the colonic microbiome is significantly depleted, usually from 750, maybe 1,500 species, down to perhaps somewhere between 200 and 400. And then that allows the C. diff to flourish, to produce its toxin, and then that causes the diarrhea, which kills people. And interestingly, one of the most effective treatments is fecal transplant, where you take somebody else's poo and you put it into the colon of the affected individual. And something like 94% of people are cured by that intervention, presumably because it then recolonizes the bowel with normal bacteria. So in conclusion, just think about this. Within the body of a mature human being, human cells have only 1% of the genes. The microbiome holds the other 99%. The interaction between bacterial products and human cells may significantly impact human cellular development and function. And through the impact on gene activity, immune and inflammatory and metabolic, and that's manifest through the impact of gene activity, immune and inflammatory and metabolic regulation. Thank you for your time, and I would now ask for any questions.
Well, Simon, that was Absolutely fascinating. Thank you very much indeed. We have, there's always a little bit of a pause before the questions come, and I've got a particular sort of personal question to ask you. As somebody who is heading into his 60s, how do I look after my wonderful biome better than I might have done already, or is it too late?
No, it's not too late. There are examples of people in their 60s increasing their microbial diversity, but you'll not be surprised to know that the best way to increase your microbial diversity are all the things that we say about general health. So in general, get enough sleep, enjoy a diet which is rich in vegetables and has enough lean protein, but you try and have as much vegetable variety as you can, take regular exercise, and all of those things not only increase your microbial diversity, but will also increase your general well-being and your likelihood of living to a ripe old age. I'll be with you there.
Okay. I mean, I have to say that I feel that with all except the sleep, I'm doing okay with that lot, which is great. I have a question from Mike Shaw, who asks, should we be making children forage in a school allotment from time to time? I presume that means getting our hands dirty. And
so I can't think of a reason why not, and I can only think of positive benefits on a number of levels. So I think that is a positive thing, because they will appreciate the benefits of vegetables, and hopefully it will broaden their diets. But I also think that it's a form of exercise. It gets people outside, and I think that's probably good for mental health and well-being. And so yeah, I would absolutely encourage that. I've been out in my allotment today. I think that that's a good thing. In terms of evidence, I can't show you a randomized controlled trial, but you know what? I can't show you a randomized controlled trial that walking in front of a double-decker bus is a bad idea, but I know it is.
Okay. I like the analogy. Thank you. We have a question from Glenn Garrett, who's watching, who says, why doesn't everyone eat Chinese mushrooms to lose weight? I can hear the word "then" on the end of the question there, if you like.
So this is a very specific fungus that encourages the growth of a very specific bacteria. And the trouble with uncoupling oxidative phosphorylation in brown fat is that you do that and it does burn more energy, but we live in a hugely obesogenic environment. So when you think of how we get fat, a pound of fat is 4,000 calories, roughly. It's just 3,500, something like that. That sounds like a lot of calories, but it's roughly 100 miles on a bicycle, which sounds like a lot, but isn't that much. It's a day's ride. And in order to get to put on a pound, you only need to eat 100 calories a day more than you need for just over a month. So say 40 days. 100 calories is not a lot. It's a banana. It's less than a whole chocolate digestive a day more than you need, and you'll put on a pound of fat a month. And what any intervention, including eating Chinese fungi, is likely to do is to marginally affect either your energy intake, what you're eating, or your energy expenditure, what you're burning. And the impact is likely to be very long term. So it's going to be a slow burn. So if this uncoupling of the of oxidative phosphorylation in brown fat burns an extra 30 or 40 calories a day, that's great, but that is all it's going to do. So it will promote weight loss, but it's a slow burn, and we live in such an obesogenic environment that you can't just take that and expect to be skinny. So it's all about balance. It may be one of the interventions that works, that helps people lose weight, but it's not going to be the answer to everything. There is no magic bullet. I'm sorry, Glenn.
Great answer. Good to see the bunch of bananas that accompanies Glenn's Glenn's icon on YouTube, which is useful given your answer. Rob Gardner, who is our glorious head of biology, has asked the question: are we looking at epigenetic changes to the human genome due to the microbiome? So something they've just started looking at in College One.
Yeah, we are.
Very topical.
Yeah, we are. We absolutely are. So so take the neurobiology example. So there is the thinking is that certain short chain fatty acids are bacterial products are making their way in the circulation to the central nervous system and influencing the switching on and off of genes within the central nervous system's cells. They've shown that the absence of the short chain fatty acid as a product is it is the thing which makes the difference. They've demonstrated that infusing the short chain fatty acid improves the glial cell function, but they're yet to show a mechanism by which the short chain fatty acid gets from the gut into the central nervous circulation, but I'm sure it will come. So yeah, it's going to be all about gene switching on and off through mechanisms triggered by the metabolic products of the bacteria or fungi.
Terrific. Amy Douglas, who's one of our College One students, asked the question: what would the impact of taking antibiotics for many years in childhood be?
That's not been really well studied, Amy, but you might. So I think it depends on the other things. So it will reduce the microbial diversity for that individual, but it's not clear at this stage because people haven't done the studies because they haven't been it's difficult to get a cohort of individuals who've done that for long enough, who've had similar antibiotics and a clean group to get the outcome of that. But the good news is that if you then stop taking antibiotics in, say, adolescence, you probably are able to increase your microbial diversity, but it may take some time. So undoubtedly it will have an impact, but don't let having an impact ruin the rest of your life, if that makes sense, because there are other things you can do about it. And you can't change what's already happened.
Right, moving on to Lara. She asks whether probiotic yogurts actually help us improve our microbiome.
That's a really interesting question. So it depends. So Yakult, for example, not wishing to mention any names, the bug in Yakult is a lactobacillus. It has been demonstrated to have health benefits. It does, so things about probiotics, you need to make sure that the bug you're taking gets to the colon. It does get to the colon. It does have a health benefit. You need to take it in enough numbers to have an actual impact on the population of bugs living in your gut. And the yogurt doesn't really have enough bugs in it. So an individual Yakult's got 150 million bugs in it, but you probably need to be taking over 1 billion to have a significant impact on your biome. So if you had enough yogurt, you probably could, but you'd have to have an awful lot of yogurt. So that's neither a yes or a no in a way. It depends how much yogurt you're eating. Certainly fermented products, things like kefir and all those kind of pickled fish that the Scandinavians bury in the ground seem to have a positive benefit as well. So fermented foods seem to have a benefit. So I think in the round, they are likely to be positive, but the impact of, 1 Yakult a day is probably small, if it's significant at all.
Terrific. I've got one last call for any more questions to come in. We'll just sort of wait and see if there's anything more comes in. When we were talking beforehand, one of the things I think I heard you say was you felt that there was a sort of Nobel Prize waiting in this particular field for someone to discover.
There is.
Can you give me an idea of why you think it's of that import, I guess, that this would lead to that sort of thing?
So I think the dream of pharma is to be able to identify the bacterial product that impacts a specific condition, so that pharma can then sell it to us for a great deal of money, potentially for their shareholders. I think that that's a nice idea in the context of a medical model, but most of the world probably can't afford that. And so I think that the Nobel is probably in understanding the computational biology of the interactions between the human environment, the human diet, and the human genome. So that actually, when somebody comes to me with inflammatory bowel disease or a particular thing, I can test their genome and their microbiome and say, do you know what? You need to do this, this, and this with your diet, which you might be able to afford and execute, or you might need to do this exercise, but whatever, in order to enable you to change the course of your disease. And in many ways, doctors have been doing that for centuries, because the archetypal picture is of the flat-capped chap in the GP surgery, and the GP saying to him, the best thing you can do is give up smoking, fried food, and alcohol. And the chap in the flat cap says, what's the second best thing I can do? So in many ways, doctors have been trying to do those things in a very gross way for a long, long time. But if I were able to say to somebody with inflammatory bowel disease, actually, do you know what? What you need to do is you need to do 100 minutes of exercise a week and take this combination of turmeric, cumin, coriander, whatever. They're not cheap herbs, I know. But to manipulate their diets in a way which was positive, that might actually be a more holistic way of tackling that. And even to get to the point where we understood all of that would have an impact on our understanding of the whole way that the human and animal biome works, and its impact on health and disease. I think that is a Nobel Prize. If you can do that, then you're very clever.
So there's the challenge to some of our students this evening. Yeah. If you fancy a Nobel Prize, there's an open path for you in that particular field. Another of those students has asked the question, when taking probiotics, should we take them before, during, or after meals?
That's an interesting question to which I don't honestly know the answer, but it's probably not critical. The other important thing is probably the meal that you're eating. So you probably want to eat a meal which, again, has lots of vegetables in it, because vegetables encourage the growth of the bacteria. And if you just live on pizza, then you're less likely to encourage the growth of those probiotics. The other thing is, interestingly, fish oils probably promote some bacteria as well. The other interesting thing is some bacteria are active at night and some bacteria are active during the day. And so the timing of the probiotics probably depends a bit on the probiotic you're taking and a bit on what you're eating. But in general, I would say the important thing is the diversity of the diet and taking the probiotic rather than necessarily the exact timing of the probiotic.
Lovely. Back to Rob Garner again, who's got a second question, which says, should people look up specific bacteria online and then buy supplements to target specific symptoms?
My straight answer to that is yes. And the reason why I say that is because, as I mentioned in the talk, the effects of bacteria are likely to be strain specific. So it's about what you're trying to achieve with your intervention. So if you've got a specific symptom, then it's about how do you address that symptom. And I'll give you three short stories. So I was having dinner with Eamon Quigley, who's the kind of great-grandfather of the microbiome. And I said to him, Eamon, I know you've got a number of strains which reduce inflammation. Why don't you, like other probiotic manufacturers do, he's a professor in Texas, was in Cork, Why don't you mix those strains and give them together? And he said to me, because we found these strains occupy the same metabolic space, and when you give them together, they knock each other out. So that's one thing which we don't always understand when we're taking probiotics. The second thing is that, as I mentioned, there are bugs that we know increase resilience in the context of anxiety. That is a strain-specific effect. We don't understand exactly what it's doing and how it's doing it, but that is a strain-specific effect. So they've looked at hundreds of strains, and that's the only strain that, as far as we know, does that. And we've also know that there are some strains which reduce the risk of recurrent inflammation in people with inflammatory bowel disease. And we know that there are specific strains that have demonstrated to reduce sensitivity of the bowel in people with irritable bowel syndrome. So you can actually take a balloon, stick it up somebody's bum and blow it up in their bottom, and they'll say at a certain level of distension, they'll say, oh, that's uncomfortable. And this particular strain means you can get more stuff in the balloon before they say it's uncomfortable. And that has been shown, and it's been demonstrated to have correlates with one or two other measures of discomfort in terms of their brains lighting up and all of that. So if you've got symptoms, take the probiotic. That's been demonstrated to address those symptoms. What you need to do is look at the science, not just look at the blurb that says, yeah, we've got a great something, it will fix everything. You probably need to say, well, have you got a randomized controlled trial to show that? And where's the science? So in short, yes, but it needs to be scientifically rigorous.
We've got another question. I think that's the last one, which has come from Mike Shaw, which says, have COVID and increased hand washing had a measurable impact on the diversity of the biome?
That's a really good question. So it is certainly true that during the period of the pandemic, the instance of gastrointestinal infections has significantly reduced. And it is likely that there has been a reduction, probably not that much in the adult biome, because that's pretty well established and pretty static, because people tend to eat the same rubbish, or rather more rubbish in the pandemic, perhaps. But in young children, it may be that hand washing has had an impact because they're still establishing their biomes, but I'm not aware that it's actually being properly studied.
Well, Simon, that's been fantastic. I feel a little bit like that character from the Fast Show that Paul Whitehouse used to play, that used to go walk around going, isn't it fantastic? Isn't it marvelous?
Yeah,
all the way through this talk.
It's amazing.
Feeling like that character, learning things which I understood as a non-scientist about the gut and its importance and the importance of the bugs within it. So I've been not just thoroughly entertained, I've been hugely encouraged and fascinated, and love to see the way that this science could have an impact, not just on our physical health, but on our mental health and well-being as well.
Yeah, absolutely.
Thank you so much for everything that you've done for us this evening. It's been a really terrific talk, which I'm sure we'll stash away in our archive, and the biology department will get out on an annual basis and show people when they're studying this stuff. So thanks ever so much for that.
Been a pleasure.
Everybody who's been here this evening. And we'll have more talks, I believe, next term. We'll let you know all about those. So thank you for your attendance this evening. And thank you again to Simon for a fascinating talk. That's made me feel quite hungry. Thank you.